Hello, my name is Grace Tieman and I am a first-year master's student in Heather Buckley's group. There are three goals for this project, and they are to synthesize cationic corals and porphyrins, to cross-link the corals and porphyrins to 96-well plate pegs and other surfaces, and to use the cross-linked 96-well plate pegs to determine if they will prevent biofilm growth in biological assays. So, here I show how biofilm growth is prevented by the porphyrins and corals. They are light activated and they release singlet oxygen. When they release singlet oxygen, it goes and disrupts the cell membrane, which then kills the cell. Here is a porphyrin that was synthesized by Stefania and the Wolf group. It is currently what we are using to figure out the best way to cross-link the porphyrins and corals to 96-well plate pegs. Here I have outlined the corol synthesis that I am currently working on. I have done the first two steps and now will be setting up the second two steps shortly. This mechanism that I am using to cross-link the porphyrins and corals to the plates and other surfaces was developed by the Wolf group. And I have also outlined how the porphyrin itself undergoes this mechanism to attach itself to the polymer. I've also included two photos of successfully cross-linked porphyrin to materials. In this photo, these are mattress pieces that Stefania successfully cross-linked the porphyrin to. And here is a 96-well plate that I successfully cross-linked the porphyrin to. This 96-well plate can now be used in biological assays. And I have also outlined how we set up the biological assays. So far we have used this methyl porphyrin in solution with the bacteria Pseudomonas aeruginosa. We have two 96-well plates that both have the same amount of porphyrin and Pseudomonas aeruginosa. One plate is left to incubate for 24 hours in the dark, while the other plate is left to incubate for 24 hours in the light. After 24 hours, a sampling of the wells are plated onto tryptic soy agar and allowed to grow for 24 hours. And then we can come back and count the colonies to see how much growth was on each plate. As you can see, all the plates that came from the dark 96-well plate have a significant amount of growth, whereas all the plates from the light 96-well plate have little to no growth. This shows that in the presence of light, the porphyrin does prevent the biofilm growth. Future directions for this project are to synthesize other porphyrin and corals to see if one of them works better than the other, and then we can make more of those, as well as determining a better way to cross-link the porphyrins and corals to the peg plates, because currently they deform, which makes the biological assays a lot more difficult. And the last thing is to perform more biological assays with the cross-linked porphyrin. Thank you for listening to my presentation.